Okay, so hello everybody. My name is Bahar ASADIPOUR, and this is the workshop at From Images to Knowledge conference, the virtual version. The workshop that is going to be on phasor analysis for FLIM data using FLUTE, which is Fluorescence Lifetime Ultimate Explorer. It's an acronym that stands for that. And it's a Python graphical user interface for interactive phasor analysis of FLIM data. This is a project that we developed at the Laboratory for Optics and Biosciences, or LOB, in the Advanced Microscopies and Tissue Physiology pole at Institut Polytechnique de Paris in Palaiseau. And I also have the pleasure to have Stringari, the person who directed this research project, with us right now. So about FLUTE. Before even giving you the outline of the presentation, FLUTE was a project that we came up with during COVID confinement in 2020. And it was the initial team, FLUTE team was Dale, Chiara, Lynn, and myself. And then Paulina joined us when we were writing the paper. The paper is published in 2023 in Biological Imaging Journal. And you can find the code and the executable on the GitHub of our laboratory. And also the FLIM datasets that we are using today, I'm going to practice with, are in the Xanadu repository that you can download. These are all the links that you need, that you can download either right now or later and work with it yourself during the hands-on session. So the outline is going to be first an introduction on FLIM, very basic, very fast, and then moving on to FLUTE and the features that it has with the applications and some examples, and then we are going to move to the hands-on session. So fluorescence lifetime and fluorescence intensity. I'm pretty sure that everybody that is here already knows what is the phenomenon of fluorescence. Fluorescence is the absorption of electromagnetic radiation at a certain wavelength and then re-emitting it at a higher wavelength. And although it's very much used and in all research domains, but there are problems with it, such as absorption, scattering, heterogeneity, and also overlapping of absorption and emission spectra that can make the identification of different fluorophores difficult for us. So this is why we are moving on to another metric for the fluorophores that is called lifetime, and is the average time that a fluorophore spends in the excited state moving back to the ground state, and is the reciprocal summation of radiative and non-radiative decay rates. Because of this radiative decay, it's molecular identification, meaning that it's intrinsic property or fingerprint of a fluorophore, and it depends on the chemical structure of the fluorophore. And because of this dependence on non-radiative decay rates, it can be sensitive to binding, quenching, changes in pH, viscosity, if there's energy transfer, and etc. And it's independent on concentration, intensity, heterogeneity, and scattering and photobleaching. So it's a very powerful tool for us to use to be able to extract information regarding, for example, in our case, metabolic information that can be found inside living cells. So the methods for data analysis can be categorized very vastly into three categories: curve fitting, fit-free analysis, and machine learning. So we are going to focus on the two mostly used ones, because machine learning is rather new. And the two other ones are going to be time domain and frequency domain analysis of the FLIM data. And the time domain, the most vastly used, is multi-exponential fitting. But there are some problems with it, such as its dependence on initial conditions and hypothesis on the sample. The interpretation can be difficult, especially in multi-exponential decays, because of the high number of fitting parameters, and it can be slow. So we are focusing on the frequency domain analysis using an approach that is called Phasor analysis, and uses a fast Fourier transform to transfer from the time space to frequency space and this Phasor space that we are seeing over here. So it's fit-free and it's unbiased representation of the data, and it shows the chemical species, the fingerprint of chemical species. So we can find them as different clusters and clouds all over this universal circle that we are seeing. It's once you get used to it, it's very straightforward interpretation of data, and we can get unlimited number of components. So it's ideal for high throughput screening. Phasor analysis for FLIM data was first introduced in Laboratory for Fluorescence Dynamics, or LFD, in the US by the legendary Enrico Grattone. And you can find excellent publications, two of which are cited over here. And it can use both time domain and frequency domain data, FLIM data, and represent the data in this Phasor space that we are seeing over here. The coordinates of this Phasor space are, and the signatures are that all the single exponential lifetimes lie on the universal circle that we are seeing over here. For example, this is the first species, this is the second one. And multi-exponential lifetimes lie on a straight line between the locations of the two single lifetimes, as you can see over here. For example, in terms of most of the experimental points that we get, especially for in vivo imaging and cellular information, they are going to be a mixture of two, according to the contribution of the two species. And so the second part is about FLUTE. As I told you, you can find the source code and the data and the publication. And it was a collective effort to have an open source code for more reproducible and user-friendly graphical user interfaces that can be used for large FLIM datasets. So the first issue to address is the data format. So as I mentioned, FLUTE performs Phasor analysis on FLIM data in the time domain. So either acquired with time-correlated single photon counting or TCSVC electronic cards, or time gating technique, provided that an entire period of the laser repetition is recorded and the lifetime decay is not truncated. For the moment, FLUTE can work with TIFF files. And every image in this TIFF stack, in this time stack, is going to represent a temporal bin of the FLIM stack in the time domain. And as you know, we have different electronic cards, and we have different commercial setups that use different data formats. And if you are using any of these systems, you can refer to the supplementary information in the paper and see how to work with. But quickly, for Becker and École, you are getting the STD files that you can export into TIFF using the plugin bioformats in ImageJ. For ISS cards, we get FBD, and using the VistaVision software, we are able to convert them to TIFF. For PicoQuant systems, we get PTU, and then we can convert this PTU to a bin file, and then the bin file to the TIFF. And for Leica systems, you can transform them. It's either PTU or bin, and you can transform them using the bioformats toolbox. So to have reproducible and reproducibility in our analysis and to have open access and easy and quantitative analysis, it's important to have different software tools. And there has been an effort in the entire community. For example, the software tool that is being used the most is SimFCS, written in C++. It is free, but the source code is not open. We have also PAM in MATLAB, which is not free because MATLAB, but it's open source. The code is open, and you can see and you can change according to your needs. And then we have FLIMJ in Java that is open source and is free. And we have the advantage, let's say, of FLUTE is that is in Python and it's both open source and is free, because like there's this shift towards more Python-based codes, and there are other there's other effort in the community with Python, such as Phasor Pi, Napari FLIM Phasor Plotter, Phasor Identifier, and FLIMPA. So in order to be able to use FLUTE, you should run main.py in Python after installing all the necessary packages. And or either you can use the executable FLUTE. And this, after like running the executable, this window pops up. On the menu, we have 3 different categories of information that we can load and we can change. In the home, you can load the calibration and your FLIM data, and you can do also bulk process. And in the interactivity data, you can interact with your data and visualize it. And in the thresholding, you can apply filtering or some thresholds. And then in this table over here, you are able to see your data. The first thing that we need to do is in the home, we should perform the calibration, and then we will apply some sort of filtering and visualization effects, such as thresholding, and we are able to, after loading the TIFF file of our raw FLIM data, we are able to visualize it in different intensity and lifetime estimations, such as lifetime, phase lifetime, modulation lifetime, and also distance from different species. And we can also perform cluster analysis using the different cursors that we have. So this calibration step is crucial, because if we don't have it, our Phasor cloud is going to be somewhere random, basically. And we are performing this calibration with fluorophores with a known lifetime, such as fluorescent, which has a known lifetime of 4 nanoseconds, coumarin, rhodamine, and you can also use the second harmonic generation signal from starch or nanoparticles, KTB nanoparticles, that has the lifetime of 0 nanoseconds. And what you need to know is the bin width, which is the duration of a single temporal bin of the time domain FLIM acquisition, the laser frequency or your repetition rate of the laser, and for multiphoton FLIM, we are mostly using 80 megahertz. You need to know the lifetime of your reference. For example, in this case, is fluorescein. And then the harmonic part is the integer multiple applied to calculate the Fourier transform. So we are using 1. After having done this, you are able to upload and load your data. And what you see at first is something like this, can be very noisy. So we are applying a convolutional median filter 3x3 to reduce the noise without having anything to do with the spatial resolution. And in order to reduce the noise, background noise, or like unmodulated light, you can use this threshold window to get rid of the noise that is, for example, over here in the background, to go from something like this to something like this. And then you can interact with your data and explore what is going on your data with different, and you can use cluster analysis with different cursors that I told you, and I'm going to show you later on. We have different lifetime estimations, as I just mentioned. We have the phase lifetime. The formula is over here. Modulation lifetime, the formula is I'm not going to go through the formula, but you can refer to the paper, and if you have questions, you can ask later. And then a distance from known molecular species. For example, in our case, we are interested in NADH, which is the metabolic cofactor, and we know that free and bound NADH have different lifetimes, and by knowing the lifetime of free NADH, we are able to calculate the distance from free NADH, from bound NADH to this species. And we have different, you can visualize these different lifetimes, tau p standing for tau phase, lifetime of the phase, and tau m standing for modulation lifetime, intensity, and the distance from free species. And then you are able to save and extract information from the files that you're saving in text and also TIFF. I see that maybe I have a question, so I'm just going to open it. Is the harmonic the same as the number of components in the multi-exponential decay? No. No, it's not. It's the integer that you apply to get your Fourier transform, if that makes any sense and if that's clear. I'll move on to the next slide. So for the future developments of FLUTE, we are interested in direct import of the common FLIM file formats, for example, STD, FBD, and PTU. We are interested in having GNS and intensity coordinates as intermediary file formats. It's always interesting to increase the speed using GPU. And also interesting to adapt phasor analysis to typical time-gated sampling limitations by taking into account the effect of decay truncation and gate shape. It's interesting to do integrate a fully automated calculation and mapping of fraction of molecules, and different to have more advanced analysis tools such as different filters and cluster analysis, and also last but not least, moving everything to Nopari, because it can be of use for the users out there. So we are approaching the demonstration. Before that, I just want to show you what the data looks like and what we are expecting. So one of the first data sets that we have is to map the distance from M-cherry to second harmonic generation response from collagen. And the data that we are having are three. First is the IRF, which was acquired using SHG, so zero lifetime. And then we have this first image, which was collected without any filters. So we have both M-cherry and collagen, and you can see the phasor cloud looks like this from M-cherry to collagen at 0. And then we have, by applying bandpass filters, we are able to see only the M-cherry, which is over here, and only collagen, which is over here. And you can see the corresponding phasor plots of intensity and distance from M-cherry. The second data set is FLIM of intrinsic metabolic cofactors, as I told you, nicotinamide adenine dinucleotide in its reduced form, or NADH. And the signature of this metabolic cofactor is that in the resting cells, we have longer NADH lifetimes. So we have free NADH and bound NADH, and free NADH has the shorter lifetime with respect to protein-bound NADH. And when the cell is in its resting phase, it's going to have longer NADH lifetimes, which corresponds to more bound NADH and abundance of bound NADH. And in the proliferating cells, this phenomenon is mostly shifted towards a more glycolytic metabolism, which corresponds to short NADH lifetime and an abundance of free NADH. And what we have is our cells, and we are seeing the intensity, tau phase, and tau modulation, and also distance from free NADH. And we are inducing rotenone to our data, which is an inhibitory of the glycolytic pathway, which shifts the lifetime towards a more glycolytic pathway. And what we are seeing over here is this on top is in the control, and on the bottom is in the rotenone treated. And what we are observing is that the phasor is shifting towards a more free NADH and a more glycolytic and a shorter lifetime. And so this is it. If you don't have questions, I would like to move on to the demonstration. Hi. For FLIM analysis, for the moment, I'm not sure, but this is something that we can take into account for later, for sure. I don't know if Kiara has more comments to add, or maybe you can address some something that I missed in the chat. So, are you able to determine the percentage of bound versus unbound NADH from your analysis? We can get the distance from free NADH because we know the lifetime of free NADH, which is 0.4 nanoseconds, short lifetime. And then we are able to get this distance from free NADH. Percentage, you can then normalize and get a percentage, if that makes any sense. Is it possible to work with FLIM time series? What do you mean, like time lapse? Time series? I don't. Meaning that you upload your entire time lapse stack into FLUTE? For the moment, no. But this is an interesting feature that can be added to the program. For the moment, you can only do one TIFF stack at a time, and but you can do batch processing, meaning if you have your time lapse instead of in one stack in one file, in separate files, like time points, you can do a batch processing, and that is rather fast. Okay, can you explain the definition of distance you just mentioned? Yes, maybe I can show you. Okay, so can you see this slide? Hang. Okay, so this is exactly the definition of what we mean by distance. So what we are doing is that we know where is the location of this free NADH, and we are getting the information based on the data that we have of our cloud, and we are able to measure the distance from that one. Okay. Okay. If you have two separated clusters of signals, mCherry in nucleus and NADH in cytoplasm, would you expect to obtain two dots in the Phasor plot? Or it's more like a large cloud, and everything is sort of bound together, probably not two separate clouds, but what you can do is to perform cluster analysis and see which pixel corresponds to what location in the Phasor. I'm going to show you right now in the data that we have, if that answers your question. Okay, so if everybody agrees, maybe we can move on to the demonstration. So we are going to reproduce what we have in the paper. Okay, so I have FLIM over here. Okay, so these are all the data that you can get, and I just want to show you what does it look like in time domain. So for example, let's put one randomly over here. It looks like this. We have 56 images, and that corresponds to temporal bins. So in order to calculate the time temporal bin that we have, what we need to do is to divide our repetition rate by this 56, and it's 12 point over 56. So it's 223 picoseconds, or 0.223 nanoseconds. And this is like to show you and this is the typical shape of a fluorescence decay in the entire image, not any specific region of interest. So what I'm going to do is first, maybe let's start with this one. So fluorescent embryo is the calibration. So before calibrating the system, we have phi calibration 0 and M calibration 1, and then I'm going to load the calibration. So this should be this, and then the tau reference, because we are using fluorescein, is 4 nanoseconds. Laser frequency is 80, and we are using harmonic 1. And what we do is that, like, we load this fluorescein embryo, hoping that it works. Okay, so calibration and phi and M calibration changed, as you just saw, and then we are able to load FLIM. And I'm going to load like this embryo first, see what we get. I think my screen is a little bit small for everything. I apologize for that. So without any filters or thresholding or anything, we are getting something that looks like this. So what I'm going to do is that I'm going to go to this thresholding menu and apply median filter, 3 by 3 median filters. So it's going to shrink the cloud a little bit, and then we are getting some sort of noise. So I'm going to put on a threshold of 20. So it's going to reduce all the pixels that have less than 20 photons. And I'm going to leave the intensity max as it is. And in the interactivity part, So as you can see, we get two different clusters that are evident over here. What we can do is that we can place a circle over here in the red part and see that this part of the image is reacting to the circle, is reciprocal. And then we can change the color. For example, let's say, let's put yellow. And we can put it over here. And we can see that the cell part of the image is responding to this, meaning that every part that is over here belongs to this cloud more than every part that belongs here is from the second cloud. And then we can clear all circles. And this is the default image that pops up is the intensity image. So what I'm going to do right now is that you can, if I bring the laser, the Phasor, what you can do is to play with this angle range, for example. And you can see that as you are moving and cutting this second cloud, we are also eliminating these pixels on the image. So I'm going to get rid of all the noise that is over there because so, and now what we can do is that to show like the tau p, and this is the lifetime map that you can see. And what does it mean? It means that these parts, the blue, this is a jet lookup table. So the blue means that it's shorter lifetime and is closer to free NADH, and this more orange red part is more bound NADH and has a longer lifetime. So this was just to show you a little bit of the cluster analysis. Moving on to, I have a question. Let's see. Okay. The comment from Yara. Okay, for example, these two images. One is control and the other is rotenone treated. And we are expecting to see a difference in the lifetime of the cells because of this inhibition. Okay, so it's good that this happened because like you see that the images look a little bit weird, and it's because I'm using the calibration from the previous image for the images that have a totally different calibration. So this is not good. I'm going to remove these and load the correct calibration for the cells that we have. And it's fluorescein, this one. So as you can see, again, the calibration changed, and I'm going to load the FLIM files. And so these are, one is zoomed. I'm going to use the one that is not zoomed. For every time that you are doing acquiring FLIM data, you have to get the instrument response function because it might be different from one day to the other. And these are two data sets that were acquired in different days, therefore different calibration file. So I'm going to select both of them, and then I'm going to reset the range. And this is already sorry. I'm going to see if I can change. I don't find the second Phasor, sorry. Okay. So maybe what we can do for this one is to increase the intensity minimum intensity that we get, to see like if we are able to get rid of more noise. Okay, not too bad. And then because I have them both selected, if I go in the interactivity and try to change the range, is going to change for both of them. And you can also do the same. I'm doing it with the tau phase, but you can also do it for the modulation, for example. If you use the second line comment, you're able to do this. And as I told you, the default image is the intensity image, so we can now change it to one of the lifetime maps, and for example, tau p, and visualize what we are seeing. But I think we can do more even. And so this is in control, and this is in rotation. Okay, I see that I have like 8 notifications in the chat. Yeah. So is there any way to demonstrate the distance measurement? I am thinking that whatever the value is from the distance measurement free NADH is the fractional value for bound NADH, but I just want to make sure that is correct. I don't know if Chiara has. Yeah, you can. You can do the map of the distance here. So we know that this is NADH information. You enter the lifetime of the fluorophore of your choice, here free NADH, so you enter 0.4. And then you're able to show, this is the map of the distance from free NADH. And in order to get the fraction from NADH, free NADH, what you have to do is, fraction of bound NADH, sorry, what you have to do is that you have to, it's all in the paper, you just have to divide over the distance that you get in order to get that value. So when you extract information in every single cell that you get, you are able to see the difference in the lifetime of these cells, as you can see in this plot over here. So for rotenone treated cells, we have a more glycolytic lifetime, and the lifetime is shorter. Okay, so if you don't have questions regarding this data set, I might move on to the last one. Okay, so I'm moving on to the last one. So the last one to remind you is the one with M-cherry and collagen. The collagen comes from a second harmonic generation and not autofluorescence. And the calibration file is the IRF from second harmonic generation in starch. So I have to again calibrate FLUTE, and this time the reference lifetime is 0 nanoseconds because it's spontaneous. It's SHG, second harmonic generation. So to choose the file, we're gonna go there, and it's called starch SHG IRF. And then these are the three images that we are interested in. So first, I'm going to do the one with no filter. So because the values that I chose before are to locate the phasor over here, we are seeing nothing in our image. So what I'm going to do is that I'm going to reset the range. And this is our intensity image. By the way, if you double click on the intensity instead of the jet lookup table, you can get the gray lookup table. And I accidentally tapped on the phasor, so now we have a yellow cluster over here, which is basically nothing. So I'm going to clear all the circles. Mapping of the free NADH. Okay, sorry, I closed the data set. I'm going to finish with this one and then come back to the other one. Okay. So yeah, but I prefer the jet, so let's go back to here. And then if I were to show you what does it look like in terms of clusters, we have M-cherry over here, and then everything that is M-cherry is going to pop yellow, and whatever is collagen is going to pop red over here. This is in the tail of the zebrafish. So in the fins, we have a lot of collagen structure, and when you image, you can collect the SHG coming from the collagen. So this is without any filters, and wow, sorry. And then if we put the bandpass filters, we are able to collect only M-cherry and only collagen. And I'm going to load that as well. So this is the M-cherry. Which has a longer lifetime. And this is going to be the SHG coming from collagen. So as you can see, it's like in this region of the phasor, we are only getting, this is short lifetime, 0 lifetime, and this part of the phasor is infinite lifetime coming from unmodulated light. So this is what we are able to see, and we are able to see the fraction of the distance from M cherry. Okay, we have some notifications. How do you know where to put the red circle? I just choose. I can change it as well. For example, I can put it over here if it reacts. You can put it wherever, and then if there is a pixel in that part of the phasor, it's going to respond to that circle and become red on the image. Can you select the area from the phasor plot with a shape other than a circle? No. What about all the pixels in the middle between the circle? You can choose them, but it's going to, for example, like allow me to choose another color. It's becoming a little bit slow. I apologize for that. You can obtain S, G, intensity, tau M, tau P, and distance from the species that you want from them. In the files that are saved, you can window tiling. What do you mean? Okay, so it's not responding to me. Do you recommend our calibration file, the fluor, to have the same excitation emission spectra as the label-free signal we are expecting from our... Yes. Especially if you're collecting with different detectors, you have to calibrate for each and every one of them, so it would be of use. Okay, so if you agree, I'm going to just close this and put back on the previous one where we can see the distance, the map of the distance from free and ADH. Oh, now it's responding to me. I have to load the calibration again. For the other data set is fluorescence, so 4 nanoseconds. I'm going to choose my file, which is this fluorescence HMSC. And then load FLIM. Yeah, let's do 60. To get rid of all the background and only get the cells. And okay, so we were interested in the distance. What I'm going to do is first get rid of everything that is outside our phasor. And then, so this is a tau p. As you can see, the cell nuclei have a higher, shorter lifetime compared to the cytoplasm. And if we put distance, so this is NADH, and the lifetime is 0.4 nanoseconds. So I'm going to like enter that, and we're going to see something like this. And you are able to extract all the information in a text file in every pixel. And if I limit it a little bit more, we are able to see this difference more in the nuclei compared to the cytoplasm. If pixels between circles provide any biological information. Okay. So I'm going to load that again. Maybe that helps for Ignacio. So this is starch. This is 0. I'm going to put starch. And then, so this is the exact same data that we collected, just with a filter, a physical filter in our detection. This is without any filter, and it looks like this because of this linear combination that Chiara just mentioned in the chat. And then without that, with the filter and without this linear combination, it's going to, the M cherry part is going to look like this. Just the cloud that is more or less tiny and has the lifetime of around 1.5 nanoseconds. Okay, I think the confusion I am having with the distance analysis is that there's no numerical value, and I am not really sure how to obtain a numerical value for quantitative analysis. Numerical value for your line, you mean? Okay, so you have to extract that from the files, the text files that you get. If you want to do that in the average area that you are interested in, or in every pixel, and what you do is that you divide your distance over that value to get something that is normalized and can give you the fraction of bound. I don't know if that answers your question. You can get the values in the text file, yes. I can show you maybe. For example, here, let's save this one. Save data. You can save either all the color maps or just the one that is already displayed. And we don't care about that. So I'm just going to do the one that it's displayed on display. Let's just directly. Okay. So this is the text file with all the parameters. We get the number of median filters used, 3. The intensity minimum was 60. So anything below 60, any pixel below 60 is not there. And then we get the the ranges that we use for modulation and phase, and then average G coordinate for all the pixels that remain. S coordinates, so G and S are the coordinates in the Phasor plot. G is X and S is Y. Average tau P and average tau M and average distance. Yes, this is the average in all the pixels that remain on our image. So everything that is here. So I'm done with the presentation and with the demonstration. If you have more questions, I can still be here a couple of minutes more, although the time of the workshop is coming to an end. Okay, to measure IRF, could we use the reflection signal instead of stars, or is it a problem? I think that you could. Yeah. That's Kara saying. Yeah. Yeah, so basically anything that has a 0 nanosecond lifetime, you can use as your IRF. Well, thank you everybody for being here. Thank you, Milena. Thank you, everybody. Okay, so maybe we can stop. I can stop share. Okay, ciao. Bye-bye.